Hello students, a very good afternoon. So today we'll be talking about we're going to cover some of the topics from biotechnology principles and applications. So today we'll start from what are the process of recombinant DNA technology, okay? So before we start the topic, so I will launch an NPS check. Please let me know if my audio and video is clear, okay? So I've launched the poll. So you just have to answer the poll to just let me know if the audio and video is clear, okay? Okay. Students, I'll just go on with the class. So if you have any problem or any doubt, any issues with the technical things, you can put it in the chat box and I'll try to resolve it as soon as possible. So let's get back to our last session. So where did we stop? So we were talking about how to do the screening of recombinants from non-recombinants or transform from non-transformants, correct? No? So during that process, we understood two important things. One is the plating method, where we try to differentiate these cells which are having our gene of interest and not having the gene of interest through antibiotic resistance characteristics, correct? And next, we started understanding what is insertional inactivation. And in insertional inactivation, we took an example of LAC-Z gene. And in LAC-Z gene, we tried to insert our gene of interest at the LAC-Z position of your LAC gene. So if it is completely occupying the gene, we understood that that means our gene of interest is in the right position of our vector, correct? So if it is not in the right position, that means it's still going to produce the beta galactosidase. That shows that the insertion is not being done properly, correct? No. So then it will result in two different types of colonies. The cells which are carrying our gene of interest at exactly the locations of LAC-Z gene, they will produce white colonies because they will not be able to produce beta galactosidase, correct, right? Whereas the blue colonies, one, even though they have our gene of interest or may not have the gene of interest yet, because of the disposition or they have not occupied the what do you tell cloning region, right? The cloning site. So that is why it will start producing the blue colonies, so which are rejected for the further use. Okay, so this we understood that how to select our cells, right, to differentiate from transformants and non-transformants, correct? So now let us summarize what is the steps and processes that are involved in the recombinant DNA technology. Okay, so first one, what do? We do the isolation of genetic material. So for the isolation of genetic material, as I said, all the living organisms, okay, have their genetic material very intact. Whenever we talk about eukaryotic organisms, the eukaryotic organisms have their genetic material even more intact than compared to that of prokaryotic organisms. So now, for isolation of genetic material, we have to undergo several, cell digestion techniques so that we get rid of all the cellular organelles and then only the DNA is left. And during this process of isolation of genetic material and then identification of the gene of interest, first of all, so there is a process, isn't it? So first we have to know what is the gene that we are trying to isolate. Next, we will go for isolating the entire genetic material. And then what do we do? Once we have the entire genetic material, the information is very, very large. Okay. So if the information is very, very large, then it is very hard for us to, exactly isolate the gene that we are looking from the entire pool that is available, correct? So what do? We try to cut the DNA, okay, in specific locations using restriction enzymes, okay? So once we have small fragments of this DNA that we isolated, correct? So sorry, that we treated with restriction enzyme. Next, what we will do, we have two things now. One is based on the availability of the gene of interest or the volume of availability of gene of interest. We either undergo gel electrophoresis after this, or we will undergo the PCR technique. Okay. So after cleaving our DNA at specific locations, next we have to undergo what you call agarose gel electrophoresis, where we separate the fragments and we isolate the gene of interest from this gene pool. Okay, the entire pool. So we'll talk about what is the principles of agarose gel electrophoresis. After extracting the gene of interest, next you can undergo the process called as PCR or polymerase chain reaction, where you make multiple copies of the gene that you have isolated. Okay, so now once we have to isolate the gene of interest. Once we have successfully isolated the gene of interest from the cells, okay, then we can make more and more copies of it outside the cell environment itself, and that technique where we make more and more copies of the gene of interest is called as PCR or polymerase chain reaction. So once we have thousands of copies of our gene of interest, now what do? We try to insert this gene of interest into a vector. So after we put it in a vector, next what we will do? We will try to insert this recombinant vector, okay, or recombinant DNA into the host cell through a vector, or through several means are there. Okay. After having this gene product, okay, after having this recombinant cell, next what we will do? We will try for what is the gene product that we are getting. Are we getting the right product, or getting the appropriate qualities and quantities that we are expecting from this particular engineering process? Okay. So once we have the appropriate gene product, next we will go for downstream processing. See, when we are trying to build a proper gene product, we will be the cells will be provided an appropriate environment so that they will divide and reproduce faster, as well as they can biosynthesize these gene products in a very large volumes. Okay. So after we have successfully completed in designing this environment for these cells, now we have the product of this entire engineering process. After that, we have to make sure that these products are available very safely for the use of for medical use, for any other use. It has to be very, very safe. So that is what is called as downstream processing. Downstream processing is very, very intense purification techniques, where it is made as safe as possible for the human consumption, okay, for any other applications. So these are the process of DNA, recombinant DNA technology. So we understood what is the steps. Let us see what is the details of the steps. Okay, first one, when we talk about isolation of genetic material. So when we talk about isolation of genetic material, as I said, you take bacterial cells or plant cells or animal tissue. Okay, so they are all having a lot of cellular organelles, right? So these organelles are made up of same biomolecules, okay, like lipids, or you have proteins, right, or carbohydrates. So that is why they are treated like lysozymic enzymes. So when they're treated like lysozymic enzymes, so these lysozymic enzymes can be proteases, amylases, okay, carbohydroxylases. So they're RNases. So when they are treated like this, for example, in plant cells, we can also use cellulase or chitinase in fungi. So this will lead to enzymatic breakdown of the cells. So after the enzymatic breakdown of the cell is accomplished, now the DNA and other macromolecules like RNA and proteins are released. So these macromolecules are again treated by the digestive enzymes, correct, right? So once they're treated by the digestive enzymes, now we have only the nucleotides, especially in the form of DNA, okay, or nucleic acids in the form of DNA, right? So once our entire solution is having only the DNA and the rest of the solvent, now what we will do? We will try to add chilled ethanol to this particular suspension. So once we have this chilled ethanol, now what will happen? The chilled ethanol will remove the excess water molecules from the DNA, so making it precipitate. Okay, so once we add ethanol, the water molecules are removed from the DNA strand. That will make the DNA to precipitate it out into a form of fine threads. Next, what we will do? We will do a process called as pooling, like this. Okay, when we spin the as we keep on mixing. So now all the DNA will clump in one area as a supernatant, like a supernatant. So we can just pull it out. So now we have isolated the DNA. As I told, when you talk about after isolation, next hour we have to make sure that this huge amount of DNA that we have isolated from our cells, we have to make sure that we have a we have a proper methodology to isolate our gene of interest, right? For example, in long stretches of DNA, we have to pick our DNA from between. So for that, what we will do, we will try to make sure that we will cut this DNA into small pieces, and this cutting of DNA into small pieces, that is accomplished by incubating our gene of interest, okay, with restriction enzymes. So when we incubate at a very particular temperature with restriction enzymes, now we have used specific restriction enzymes because we know where we want to cut, so that we will have our gene of interest of this particular base pair length. Okay. So now when we add the restriction enzymes, now what will happen? The DNA will be converted into small fragments, like DNA fragments. Now, when we have the DNA fragments, since that we have an idea that where we are trying to cut, and that is why we have used specific restriction enzyme. So now what will happen? This would have given us an idea that my gene should be this base pair length, this much big base pairs. So using the idea about how big is my fragment or how big is my gene of interest should be, then I will undergo one more technique called as agarose gel electrophoresis. We'll discuss this in detail. So in agarose gel electrophoresis, now what happens? Based on the size of the fragments, okay, they are separated out, okay, on a gel membrane. Okay, so as we separate these fragments, now we will be able to understand where our fragments are hybridizing after they migrate. Okay, we'll talk about this, like what is the process of agarose gel electrophoresis. So based on the size of the fragments, our fragments will be separated, and from that particular gel, we can isolate our gene of interest. Okay, so let us see what are the steps involved in. Okay, we have it. Yeah, we have it. Once again, what are the steps that are involved in agarose gel electrophoresis? See, first what happens? We have to treat our gene of interest with restriction enzymes. So once we have the fragments of the restriction enzymes, our next step is to prepare, okay, an agarose bed. Okay, so when we prepare an agarose bed, it is actually we specifically pick an agarose gel bed. Why do we do that? It is because when you see a sieve, for example, in kitchen and all, we'll use to separate the grains from the sand or any stones. So for that we use sieve, right? So in this sieve, if you see this pore size or the pores in the sieve, like for example, you will filter the tea, right? So if you see the sieve that you use for filter, is it of different sizes? No, right? They are very uniform in size. They are very cleanly arranged. Similarly, when we use agar-agar or agarose gel electrophoresis or agarose gel as a medium for electrophoresis, so this agarose gel, their arrangement of molecules, I mean like the arrangement of agar-agar molecules from one agar molecule to another agar molecule is very uniform. So since it is very uniform, it will create a very beautiful sieving effect. Understood? So the sieving effect means their intracellular intermolecular space is very even throughout it. Understood? So that will make it a very ideal sieve for the process of gel electrophoresis. Understood? Now what we will do, once we have chosen this agarose gel, okay, so or agar-agar, so we will prepare an agarose gel, and then we will make sure that we will pour this agarose gel onto a mold. Okay, so just like how you make what do you tell, molds, right? For example, for some festivals we will try to pour hot sugar mold onto some wooden things, and when we open it, we will form sugar candies, right? So when we have a plastic mold and we pour this liquid gel, the liquid gel will cool down and it will result in a solid agarose bed. Okay, so this bed is very important. Okay, that will only give us the sieving effect. So now what we will do, we will make sure that our bed, I mean like the entire agarose gel is evenly mixed with a strain called as ethidium bromide. Okay, And then we will try to place this bed onto an agarose gel electrophoresis, okay, device. So here what happens? So here is our, wait, let me see if you have, yeah. I think we don't have. Anyways, so what we will do, we will have an agarose bed. So we will try to make sure that we have wells. That means we'll make small holes on one end of your agarose bed. Okay, understood? So now what we will do, we will try to label our DNA. Okay, we will try to make sure that our DNA is labeled with any, for example, fluorescent dye or radioactive dyes. So for example, we are using ethidium bromide. It is one of the dye. So when we are exposing our DNA fragments to ethidium bromide, now the ethidium bromide molecules will go and attach us to the sugar backbone of your DNA. So that will make us visible, okay, whenever we try to flash an UV light. So because the ethidium bromide have the characteristics of fluorescence, it will show bright bands, okay. So we want to make sure that we can see the fragments that we are separating. Okay, we will label it with that. And next, what we will do, we have made a well, we have labeled our DNA fragments, and then we make sure that our agarose gel bed, okay, is filled with a buffer. Okay, so this buffer is very essential for us because this buffer enables, okay, the very even separation of the DNA fragments when an electric charge is applied. Understood? So when our DNA fragments are loaded onto this well, so now what will I do? I will make sure that I will pass electricity. Okay, I'll pass electricity into this agarose bed. So how do I do this? So now my DNA is, if you remember this, my DNA is what charge? My DNA is negatively charged, correct? No. So now what will I do? I will load my DNA onto the negative point, and then I have another region that is positive terminal. So I'll pass the voltage, okay, high voltage of electricity I pass. So after certain times, based on the size of our DNA fragments will start moving to the opposite pole, that is the positive charge. Why does this happen? Because when an electromagnetic field is applied, okay, so usually the charged particles move towards the opposite charge, correct? No? So that is happening. So as it moves towards its opposite charge, the movement of these fragments is based on its size of the fragment. So after the exposing it to a specific voltage for a specific period of time, we'll be able to figure out how much distance my DNA fragment could have migrated. Understood? So after we have our bands that are migrated, now what we will do, we will try to do autoradiography. Autoradiography is you will expose your DNA, okay, you will expose your DNA onto a UV light. So when you expose our DNA fragments onto the UV light, you can see that you can have the bands like this, no? Now, next what you will do, you safely take this agarose bed and try to put a nylon membrane, okay? So when you put a nylon membrane and you do a blotting, that means you take out the excess water in your agarose bed, okay? And then you will do something called as, Southern blotting. So in the Southern blotting, the DNA fragments that are present in your gel bar will get transferred onto a nylon membrane. And how do we do Southern blotting? As we keep on adding up weight, so the we place this nitrocellulose membrane on this particular gel bar like this, okay, and then what we will do, we will try to dehydrate it by addition of weight, just like printing. You add more and more weight gradually. Now losing excess water molecules in the gel, now these fragments are transferred onto a nitrocellulose membrane. Next, what we will do, we will try to hybridize this particular membrane, this nitrocellulose with an appropriate probe. Now, why do we do this hybridization? To make sure that where our bands are present, okay, which which region my gene of interest is present. So once I have this region of gene of interest, then isolation of my gene of interest is very easy. Okay, I can isolate from the pool of these fragments where my gene is located. Understood? So fine. Now I have my gene of interest from what process? Agarose gel electrophoresis process. Now this is introduced into a vector, or it is going into a polymerase chain reaction, and after that you can just make sure that it goes into a suitable host through a vector like plasmid DNA. Then you have your recombinant host. Okay, fine. So now when we see the transformation of cells, okay, how do we transfer the cells? We understood that we can insert our vector like plasmid into the cell, okay, like a bacterial cell, and make sure that they carry the gene of interest, correct? No? But if you take an example of plants and animals, grown plants and animals, you take, for example, human beings. If I want to make sure that a specific gene had to be inserted into a fully grown human being on a specific organ, let us say that I want to insert a particular gene onto my mucosal membrane, correct? No, we have mucous membrane on our respiratory, digestive, and urinogenital tract. So for some reason, there is a problem in my mucus formation. So that is why what I will do, especially in my breathing, respiratory tract, I want to insert a specific gene onto the cells that are lining my respiratory tract, so that I will produce a very healthy mucus and it helps in my respiration. Now, how do I do this? How do I insert a healthy gene to the cells that are already matured in an adult, in a matured organism? That is not possible, right? There are thousands and thousands of lakhs of cells are there, and individually going and delivering this gene, to every cell, it is very impossible. Same you take about plants. The plants which are prone to some of the infections from the, insects or any of the rodents, etc. So they are, when they're prone to it, how can you change their characteristics? How can you make them resistant to a particular pest or insect? So it is not possible to deliver the gene for every single cell manually, right? So we need to have some tools like this. So what do? We try to understand the disease, some of the diseases, and we take advantage of these diseases. So some of the pathogens, when you see how they infect our plants and animals, they try to deliver their infectious gene into our cells. For example, you have Agrobacterium tumefacium, okay? Agrobacterium tumefacium is a bacteria. Now, this particular bacteria, what it will do, it will try to inject its tumor-inducing gene, okay? It has a gene called as tumor-inducing gene. So it will try to induce or inject this tumor-inducing gene into the plant cells. So whenever the particular organism, particular plant is infected by Agrobacterium tumefacium, now you can see that some of the root nodules or stem, they become tumefacious, or they will have tumors in their roots, or there is a huge growth of these root nodes, or etc., something like that. So now what we can do is, if we are able to make sure, instead of Agrobacterium tumefacium delivering its tumor-inducing gene, we can make it to deliver our gene of interest. Anyways, it knows how to infect, or it is very compatible in infecting our plant cells. So now why not? So easily it can deliver our gene of interest without any problem to our plant cells. Correct? Now, did you understand what is a mechanism that we are doing here? Take, for example, retrovirus. What do retrovirus will do? Retrovirus will take the, for example, if you take your bacteriophage, what it will do, it will try to go and infect a bacterial cell. So when it tries to infect a bacterial cell, it is trying to put its gene of interest. No, viral DNA will get into the bacterial DNA, and the bacterial cell will be taken under its control. So instead of bacteriophage injecting its gene of interest, we will make the bacteriophage to inject our gene of interest. So can we deliver like this? So that is why in plants we use Agrobacterium tumefacium as a tool, okay, for as a pathogens, like the tools to transfer the gene of interest. Now, when you talk about animals, we use retroviruses as the tools for delivering the genes in animals, okay, in higher animals. Fine. So now, if you look at the details of this, the Agrobacterium tumefacium, as I said, they usually pass on the tumor genes. So instead of this tumor genes, if we are able to give our gene of interest, now what it will do, it can deliver it to the plants, isn't it? So you can see here what it does. So they have tumor inducing plasmid. So that is tumor inducing gene. So when they infect the plant roots, see what happens to the plant root. You can see that there is a crown gall, or the disease which called the tumorous diseases, that is caused in the plants, correct? So that is happening here with Agrobacterium. Now what we will do the same way when it comes to the retroviruses. So what do retroviruses will do? Retroviruses will actually inject their retroviral genes into our host cells. So instead of making our retrovirus inject a retroviral gene, so we'll make them to insert our gene of interest, correct? No, so instead of them causing cancer, what they're doing is they're helping us deliver our gene of interest to the to the specific host cells. Fine. Actually, we have discussed in detail about agarose gel electrophoresis, but we will just brush through the process of this because it's an important process, and slides were not available. Maybe it was not properly delivered. So we'll just brush through this, okay? So as I told, what is agarose gel electrophoresis? So this is a separation method of DNA or any macromolecule based on its size, correct? No? So the size expulsion method is achieved by a uniform matrix called as agarose matrix, which is extracted from a red seaweed. Okay. So now when an electric field is applied, so usually the fragments are separated based on its size. Because of the size is very shorter, the smaller fragments will move faster and migrate to a very large distance, whereas the larger fragments will move to a very short distance. Now, what is happening in agarose gel electrophoresis? Usually, this principle, we have our DNA, correct? No, our DNA fragments, which are generated by restriction enzymes. Now, what we will do? We will try to make sure that we break down this DNA into small fragments ranging from 500 base pairs to 30,000 base pairs. Now, what we will do? We will try to establish an electromagnetic field, okay, electrostatic field. So when we establish an electrostatic field, these DNA molecules will pass through this very porous gel. As it passed through this very porous gel, okay, now because of the arrangement of this agarose gel, it makes it a very beautiful sieve for the separation of our DNA molecules. Now, based on the size, and it will carry a specific charge and also a specific shape. So now it will start migrating and separating as bands. Now, what is an equipment? So we have an agarose gel electrophoresis device, that is a machine, okay, a small device. So you can see here there are two, okay, 2 points, okay, 2 electrodes, that we have positive terminal and we have negative terminal, okay. So now you can see this is how your DNA is loaded by this micropipette, okay. So this is a scale or to make the wells and everything. So let's see what is happening. First step in our agarose gel electrophoresis, as I said, we have to make sure that agarose gel is prepared, and then cooling. Okay, we try to cool the agarose gel, and then we'll make sure that this gel is very uniform without any bubbles or something like that. Next, after that agarose gel bed is prepared, we'll make sure that our DNA is labeled with ethidium bromide, which is actually glows when the UV light is exposed to it. Okay. Now we will pour this agarose gel onto a tray so that will make it to take a specific shape. Correct? No. Now what we will do, we will try to make sure that the agarose bed has wells for the loading of our DNA. Do we have that? Okay. No, it's not there. Anyways. So once we load the DNA, we will try to fill it with buffer. So after we run the gel with a constant voltage for a specific time, now the separation occurs. Now on exposure to the UV light, what will happen? You can see that the bands have been separated, and these bands can be isolated by your Southern blotting, and then hybridization techniques will result in you accessing these fragments that you're looking for as a gene of interest. Okay. So as I said, these are the following steps in agarose gel electrophoresis and how the movement of DNA happens based on its size. That's what they're telling. The smaller the DNA it is, the faster it moves, right? And it moves the larger distance. So you can actually calculate, like, if this is the size of the fragment, and if I am applying this much voltage for this much duration of time, so you can easily say, like, okay, this should be the ideal size of this fragment I'm looking for. Okay, so that is how we can easily understand, okay, this is where my fragment must be located. Understood? Fine. So now we have our gene of interest, correct? No, we have our recombinant DNA, and let us say that our recombinant DNA is also engineered in the form of plasmid. Okay, we have put our recombinant DNA in a plasmid DNA, let us say that. Okay, so now how do we make that? our cells, our bacterial cells, how do we make them to take up our alien plasmid DNA from its surrounding? How do we do it? See, people, when we are trying to do genetic engineering, what did we do? First, we do isolation and we purification, and then we try to add restriction enzymes. And then once we have our gene of interest, next, what we will do with gene of interest? We will try to make sure that we put this gene of interest into a vector, correct? No? So how did we do? We took this gene of interest and we tried to make sure that we put this gene of interest onto a plasmid DNA. So we call it as recombinant plasmids. So this recombinant plasmid, okay, which is having my gene of interest, correct? No, it has to get into a bacterial cell, correct? If it has to get into a bacterial cell, how does it get in? Are we trying to put this plasmid DNA manually into this bacterial cell? Or do we have any methodology to make sure that the bacterial cells itself will take up this foreign DNA, okay? Or your, foreign DNA we can tell, or alien DNA from the surrounding. So that process is called as making your host. That means the cell which is taking your, recombinant DNA, that host cell will make it as competent. Okay. So what is making a cell competent? So because of the nature of the DNA is hydrophilic molecule, correct, right? So since it is hydrophilic molecule, the plasma membrane of the host cell is hydrophobic. No, it is both hydrophilic and hydrophobic, but we know that the inner membranes are hydrophobic. That means they do not allow our hydrophilic molecule to enter. So that is why our DNA cannot pass through this cell. So when we make the bacterial cells competent, now that will lead to certain changes in the plasma membrane, which allows charged molecules like DNA. Okay, now DNA is a charged molecule, correct? It has a specific charge, negative charge. So that is why that will allow our DNA to move into the bacterial cell. So how do we do this? First, the bacterial cells are treated by a positively charged ion or cation, okay, like calcium. So whenever the calcium is added into the culture of the bacterium, now the actively this calcium is taken up by this bacteria. Every bacteria fights for the free calcium ions that are in its surrounding. So they are taking up very, very fastly. So once they are in the process of taking up the calcium, now immediately the cells are transferred onto an ice. Okay. So we incubate the cells with the recombinant DNA on an ice. So once we place these cells on ice, and next after that, we briefly place these cells at 42 degrees Celsius. Say first we are cooling the cells and then immediately we are transferring it to heating, right? 42 degrees Celsius. So now this will create very faster change in the temperature, right? So that will lead to something called as heat shocks in cells. So when the cells are at heat shock, okay, and again, when it is put back on the cells, now the bacteria, what it will do, the membranes, right, the plasma membranes, for a period of time, they will become porous. That means all the charged molecules, as the plasma membrane becomes porous, it starts allowing these charged molecules to move from outside environment to the inside environment. So this enables the bacteria, okay, to take up the recombinant DNA from the outside to inside. Okay. So whenever we make the cell competent and then we, make the cell undergo heat shocks, so that will create a change in the plasma membrane that leads to the temporary opening, okay, of pores and the plasma membrane that allows these charged particles like DNA to pass through. Okay. So that is happening. So next one, you can see here, this is the process of how we make the cell competent by adding calcium, like in the form of calcium chloride. So now we have added plasmid DNA, and then first we go cool it, and then again we put it in the heat shock for 42 degrees Celsius for 2 minutes. And then again, when we put it in cool, now you can see that the DNA, which is outside, the plasmid DNA, which is outside, has come into the cell. Okay. Now the bacteria becomes transformed, as simple as that. And another technique is called as electrophoration. Okay. So in electrophoration, you can see here, what do we do in electrophoration? We have our plasmid DNA, correct, outside, which we have to put it in the bacterial cell. Now, a very brief electric shocks are produced or pulse is produced. Whenever a pulse is produced, now as I said, because the plasma membrane is hydrophilic as well as hydrophobic, and hydrophobic is in the interior, that is why it does not allow hydrophilic molecules like DNA, or plasmid to easily enter. So whenever an electric pulse is provided or generated, now what will happen? There will be a brief change in the plasma membrane that will make the plasma membrane to have pores. Okay. So these pores will allow any charged particles which are outside to come into these pores, similar to that of what happened, in making the cells competent. Okay. So these pores will seal back the movement, this charge. Okay. After this, plasma membranes will regain back its polarization. So now you can see that the pores have closed and the DNA has already been inside. So you can see in detail what is happening in electrophoration. So what is happening in electrophoration? The host cells, okay, and the selected molecules, whichever it is, whenever an electric circuit or electric pulse is passed at optimum voltage for few microseconds only, okay. So now what will happen? This will disrupt the phospholipid layer. That means it will make it to open the phospholipid layer temporarily as pores. And these pores will allow any charged molecules like DNA, okay, to drive from outside to the inside of the cells. Okay, like this manner, you can move your recombinant plasmid into your host cell, correct? No? Very easy. So we have two methodology, correct? And this is called as electroporation. And we have other methods. What are these other methods? Some of the manual methods that we have is microinjections and biolistics, okay, are some of the methods that we use. Let us see what is microinjection. See, when it comes to genetic engineering of very important cells, okay, let us take about animal embryos. Whenever we want to work with animal embryos like human embryos or any organism's embryo, and if you want to do recombinant DNA technology, now we do not rely on bacteria or viruses or something like that to transfer our gene of interest. Why? Because the number of cells are also very less and the importance of cells also is very, very high. So that is why what we will do in microinjection. In microinjection, we will try to make sure our recombinant DNA is directly injected into the nucleus of the cell. Okay. So like literally the cell is locked in place and through the micro syringe, the DNA is transferred into the nucleus. So this DNA gets incorporated into the host DNA and then it will become recombinant cell or transformed cell. Whereas when it comes to biolistics or gene gun, so this is a very disruptive method, I can tell. Why? Because the cells are actually bombarded at a very high velocity. Okay. So that is why what we will do is we will try to make sure that our DNA is coated by tungsten or platinum or gold, which are very, very noble metals which do not react very easily. Okay. So whenever we have the gene is bombarded, or for example, you have your you have your cells, let us say, okay, plant cells. Basically we use plant cells for this because they have a very thick cell wall. No. So now what we will do, we will make sure that we bombard these cells at a very, very high velocity onto these cells. So when we bombard it at a very, very high velocity, now the DNA is protected with the metals like nanoparticles like you have gold, tungsten and all. So once it is bombarded, it goes into the nucleus. So there what will happen, the DNA gets free from this gold or tungsten coating and then it gets incorporated into the plant nucleus. So this is another method. So this biolistics cannot be used on animal tissues. Why? Because the animal tissues are very, very sensitive. So if you expose your cells to biolistics, animal cells to biolistics, the cells will explode. Okay, it's not possible. So whenever it comes to animal embryos, we go for microinjection. So the success rate of transferring our recombinant DNA into the host is 100%. Same goes with biolistics. Biolistics also you can transfer the genes with very much high accuracy, but you cannot apply the biolistics for all types of cells. Why? Because of its very disruptive method, only the plant cells with a very thick cell wall can withstand this particular force. Okay. Next. Disarmed pathogen vectors. Okay. So when we talked about disarmed pathogen vectors, we took the examples of Agrobacterium tumefacium. No. So that is another methodology to transfer the gene of interest into the host cells. Okay. So you can see here, this is my gene gun. Okay. So the gene gun, and this is also micro injection. You can see how the cells are, made porous. The cells are injected by the gene of interest. Specifically, they make holes on the holes on the leaves of this plant. See what are they doing here? They will make sure that they load your pathogens. For example, Agrobacterium tumefacium carrying your recombinant DNA, and they will purposefully infest this Agrobacterium tumefacium onto the surface of the leaves of the plant. So when they infect it purposefully, now you know that this Agrobacterium is carrying your recombinant DNA. So that will help in what? That will help in delivering more and more of this gene of interest into the plant cells. Okay. So these are the methodologies in which the gene is transferred when it comes to, plant cells and animal cells. Okay. Okay, students, are you understanding? Did you understand today's session? I mean, like some of the topics that we are discussing now. If you have any doubts, you can put it in the chat box. I mean, like, with respect to any concept, if it's not very clear for you, we can discuss it again. Okay. So we have around 6 minutes. So we'll try to talk about PCR, okay, polymerase chain reaction, which is a very, very, very important tool in today's molecular biology. I can tell. Why? Because PCR is enabled to, for us to even catch the criminals who are very, very smart. Okay. So I will give you an example of PCR. Okay. So if you take an example of PCR, imagine that, okay, there is a simple story. So in forensic labs, okay, when we visited, we asked a question. For escaping the crime or to catch a criminal, how much of DNA is required to catch a criminal? Okay, we thought at least 1 gram, 2 grams, or 1 cell at least is required to catch a criminal, right? We will think like that. But when we ask these people, forensic officers, okay, who are working for government, obviously, so they told very astonishing fact. If you want to catch a criminal, all you need is just 53 picograms. Okay, picograms. Only 53 picograms. P is small, right? Yeah. Only 53 picograms of DNA is required to catch a criminal. Now, this is very, very surprising. Correct, right? Picograms are extensively small. So you can't even imagine the size of this genetic material. How are they able to catch a criminal with only very small amount of genetic material? So they are very confident of catching them because we have a tool called as PCR, that is polymerase chain reaction. Take, for example, the COVID-19 also was done by RT-PCR, no? So there what we are doing a technique called as gene amplification. That means we are making more and more copies of the gene that we are specifically looking for. And we do it in a cell-free environment, that is in vitro. We don't have any cells. Okay, that is done in a machine like this PCR machine. So we make thousands and thousands of copies of the gene of interest. Okay, so this is called as polymerase chain reaction. Understood? So for this polymerase chain reaction, what we are doing, we are using DNA polymerase. Like you remember from the replication. Yeah, we use DNA polymerase. We use nucleotides, and we make sure that our DNA polymerase as well as our nucleotides are protected by very special enzymes. And then we run this process. So as we run this process, the resultant is we will get thousands of copies of them. So let us see what is the steps that are involved in PCR. So in PCR, it involves 3 important steps. Okay. The first one is denaturation. Second one is annealing. And the third one is extension. So let us see why we have to denaturate. Whenever we have a gene of interest that has to be amplified, this gene of interest that we want to be amplified or region that has to be amplified will always be in double strand. No, the gene will be double-stranded DNA. So this double-stranded DNA, we want to make it into single-stranded DNA. Understood? So to make it into single-stranded DNA, we subject this DNA, okay, into heat. That means we raise the temperature of this DNA. I mean, like where the DNA is present. When we provide heat to this DNA, the hydrogen bonds between these two double strands will become unstable, and the strands will get separated. Once the strands get separated, you can see that they will become single strands. So this is called as denaturation. Once the denaturation is complete, where we have separated our double-stranded DNA into two single-stranded DNA, next the immediate step that we'll do is, as the cooling is happening, okay, as the cooling is happening, we would have introduced, okay, a small synthetic primers. That means we will only construct these primers. What are the primers? A very short complementary sequences. For example, if your gene of interest has a sequence of A, C, G, G, A, okay. So our primer, what we are introducing, a very short primer, will be very complementary. For example, G, C, C. So like this, okay. A very small primer, synthetic primers are introduced, and it is cooled. As the cooling process is taking place, our primers will occupy, okay, complementary positions onto this single strand DNA. And this process is called as annealing. Understood? So now, after annealing is done, the DNA polymerase is added for extension of this DNA strand. See, you can only see that your primer has occupied only a part of your DNA, right? Now, what your job is? Your job is to do extend the rest of the DNA. When you extend the rest of the DNA, you have new double strand DNA. Did you understand the knack here? So now what are we doing here? After annealing, once we have our primers occupying the complementary positions, we will try to treat it with Taq polymerase, okay, which is a special type of DNA polymerase. Why do we call it as Taq DNA polymerase? Because Taq is Thermophilus aquaticus, okay, which is a bacterium which is really which can survive in a very hot temperature, like hot springs, okay. So there the temperature is very high now. Even though the temperature is very high, it is able to protect its genetic material without getting denaturated. So that is why we treat it with Taq polymerase, okay, and then we add deoxyribonucleotides, that is DNTPs, so which are the monomers. So when we add Taq polymerase and DNTPs, since the extension process increases temperature a lot, we use a specific type of polymerase, that is Taq polymerase. Now in the extension process, since our DNA polymerase always require a free 3' end for the extension process, already our primer is giving free 3' group. In extension process, what is happening? Our Taq polymerase will start expanding in 5' to 3' direction. Now, once we have extension, we have our one more fresh copy, first copy of our like Xerox copy, or a first copy of our gene of interest. Now imagine the same step: denaturation, annealing, extension, denaturation, annealing, extension, going for several cycles. So if you do 30 cycles or something like that, you can create up to 1 billion copies or something like that. Did you understand? So this is about the steps in a polymerase chain reaction. So we will just revise this polymerase chain reaction in the next class, because the time is up. Okay, we'll end the class here and we will try to revise it in the next class. Okay, fine, students. What I will do is, I'm not able to understand if the class if you're able to grasp the class or not properly. Again, okay. So please try to interact, because it will be helpful to understand your abilities, how it's happening. So anyways, I will provide you an NPS form again, people, so don't mind. Take a short time and try to fill it up. Okay. Students from the YouTube, you can put it in the chat box if you had any doubts, so we can carry it to the next class and make sure that it is solved. Okay. Okay, students, we'll carry out in the next class. Thank you very much for the session. I'll see you in the next class. Happy New Year.